Hello everyone. In this tutorial, we are going to talk about PCR, and the goal is to understand PCR, how do we run PCR, and what does it take for us to have a successful PCR reaction. So in front of me here, I have a printer, a picture of papers, a book, and a couple of inks. And probably the question that you all have in mind is, what does this has to do with PCR? So by definition, PCR is known as polymerase chain reaction. And it's one of the most useful assays that we have in molecular biology, especially when it comes to amplifying a particular segment or region of a DNA. So if you have a specific region such as this, which is your DNA region, and you want to amplify or make more copies of this red region, what you need to do is then to perform this PCR. But the way I look at it, the PCR reaction is quite like, not necessarily the same, but more like a photocopy machine process. Because what do you have when you want to have a copy of a specific region in your textbook? You put it in a machine, and this machine, which is known as the photocopy machine, you're going to put some inks there and some blank papers as well as your book, and you submit or you present the page or the section in which you want to make more copies of, and depending on what you choose at the end of it, somewhere here in the output, you are going to have multiple copies like this of that same region. So having said so, your PCR works more or less like this. What you're going to have, your DNA region, which is going to be like your template here, your textbook, and you submit this specific particular region. You tell it, okay, let's amplify this region. Then of course you need to give it something to work with, but unlikely you're not gonna give it like inks and blank pieces of paper. We're going to submit some enzymes and some template nucleotide and buffers, and at the end of it you should have something like this. Now in that reaction, multiple multiple genes that you actually amplified there. So that is how PCR works in its simplest form. But of course, what we are after here is not necessarily this simpler illustration. It won't be enough for us to get it, but at least it will lay a nice foundation as we dive deep into this PCR definition or work. Now, various components are required for us to run a successful PCR, as we mentioned earlier, just like when it comes to making a photocopy. But what you need here is actually your DNA sample or your target region, which is this red part here. I'm going to actually redraw the red regions here, and let me just put the blue ones as well on the side, just to show you what is going on here. Okay, then the next thing that you also need is primer pairs. Okay, so with primer pairs, these are single-stranded segment of DNA that are actually complementary to the start and the end of the region that you want to amplify. So those primer, we're going to talk more about them, but I'm going to represent them in green. The single-stranded that are going to be, this is a double-stranded piece of DNA, by the way, and they're going to be complementary to this region here at the very beginning and another region here also at the end, and we refer to them as a forward, and you're going to have your reverse primer as well. So they always work in pairs. And you have PCR buffers, and the buffers are important to make sure that the conditions are stable. And you need your free nucleotides, which are also known as dioxynucleoside triphosphate or DNTP. And of course, you're going to have DATP, DCTP, DGTP, and DTTP, and a heat-resistant DNA polymerase. And at this point, I'm going to assume that we are all comfortable with the role of polymerase when it comes to DNA replication or just DNA elongation. At large, polymerases, they play quite an interesting rule thing. So what is going to happen as soon as you have this, you are going to have a polymerase that is going to actually recognize this region like this. And what is going to happen now is that once this region have been recognized, it's going to synthesize new chains like this, and this reaction, just like your photocopy, is going to repeat maybe 33 times, maybe 30 times, however long you want them to repeat as well. And next, once you have this, let's just focus a little bit on the role of these specific reagents and why do we need to actually use them. So within a PCR reaction, looking at the roles of all those reagents, what we're going to notice is that all of them are more or less equally important, and the fact that they have to all come together is actually something that does not just happen by random. What one needs to do when it comes to this is to actually make sure that we know exactly why we're adding each and every one of those reagents. So now just let's have a look at the specific role. The first one that we're going to talk about are your DNTPs, or your dioxynucleoside triphosphate. They come into 4 variety, and of course each of which is going to use a different DNA base, like I said earlier: adenine, cytosine, guanine, and thymine. Of course, once you have your region like this, let me just call a couple of base pair, and in green maybe, or in red if we keep with a color, you need to amplify this region. So what is going to happen is that your polymerase is going to go and fetch DTP and add it here, and DC and A and G and C, and as well as A here. Don't mind this B here. So that's why this nucleoside dinucleotide triphosphates are really important, and during an extension phase they are actually going to allow us to incorporate or to elongate the chain that we are working with. Next is the role of primers. Primers are quite crucial, and they play an indispensable role when it comes to this elongation, because what we're going to notice is that they hybridize with the DNA sample and specifies the region that we need to amplify. So in this gene that we have, again like this, I'm going to re-represent this. This gene could have a region that starts like this and so forth. Now when we add these primers, what they do is that they are actually going to bind somewhere around the beginning of the region of interest. Please keep in mind that this is a full strand of DNA, a larger sequence, but you're only interested in this overlapping red region. So these primers, they actually allow you to bind, and what they do, they guide polymerase, or they tell polymerase in its simple form, hey, this is where you need to start the reaction. And polymerase will be, all right, let me start here, and let me start here, and I won't waste my time here or here because you didn't tell me so. So that's how this primer work as well. And usually the length is really important. That's also something that we need to pay attention to, and they should vary between 18 to 25 base pair. Of course, some may say 15 to 20, but still it stays within that range. And you may synthesize this or make this actually in your laboratory, or you can actually order them by just giving a biotech company the base pair that you're interested in. And next you have your polymerase, and most of the time you see we always refer to it as Taq polymerase. This polymerase is quite different from most polymerase, in fact, that bacteria and every other species have, because this is a thermostable polymerase. This is important because a PCR reaction, from its basic definition and principle, it's actually just a repetitive step where we constantly breaking or denaturing DNA by opening these strands. And once we've opened them like this, the next step is we actually add more, expand them, and close them. And this process of denaturation, which is breaking these hydrogen bonds in the double-stranded DNA, needs an increase in temperature. And if there's one thing that we need to know about enzymes, is that at high temperature, if that enzyme is not comfortable, it's not going to function. In fact, it may even become less functional or denature. All right. So our process, usually, as you see in the later stage, requires quite an increase in temperature. And this polymerase, known as Taq polymerase, actually was discovered from a bacterium known as Thermus aquaticus, which was discovered in hot spring, and they seem to be comfortable in a temperature that is above 80 degrees Celsius. So because of that, the assumption was that, and that's just the start. From 80 degrees Celsius above, they're very comfortable. The assumption is that the polymerase should actually be fine and continue to synthesize new DNA strand at higher temperature. And that's the reason why we use those polymerase coming from this Thermus aquaticus, and as a short term, we always refer to them as Taq polymerase. And last but not least is PCR buffers. Most of the time we see magnesium chloride, but you can also use potassium chloride or the 3-HCl. And this is just to make sure that the condition for our reaction are actually optimum. All right. And at the end of your preparation, what you should end up with in a lab is a very nice AP tube like this with all your ingredient or reagent all together, a Taq polymerase, a primer, a bunch of primers, the DNTP, your buffer, and your DNA strand, and you put it in this nice PCR machine, and you set your temperature for the 3 steps that we are going to talk about next in this video, as soon as I get to clean my wonderful screen here. So now that we're comfortable and we understand what goes into PCR, what we add and all that, what we should look at now is what are the specific steps. Okay, having the ingredient and all that, it's nice and it's good, but at the end of the day, we need to know the steps now. So basically we have 3 steps. The first one is strand separation, also known as denaturation, because whatever we're doing there is just opening the strand, the DNA strand, like this. Then after that, you have hybridization of primers. Once you've opened the strands, you add your primers like this. One is going to go here, and if I put it nicely, one is going to be here and the other one is going to be here. Then you have your other step, which is going to be DNA synthesis. Out of those primers like this, we are going to make a new strand of DNA. So if I redo this, I'm going to add in blue. It's going to be my new piece of DNA here and here. And we just repeat this over and over and over. Technically, the steps are denaturation, where the template is heated in such a way that the 2 complementary strands are separated. Then you have annealing. The temperature is decreased just to allow for the attachment of the primers. And your third step is going to be your extension. Once the primers are added, your Taq polymerase will take its position and will start producing new strands by fetching those DNTPs. And pay attention to the temperatures as well. As you can see, you have 94 degrees Celsius, between 55 and 65 degrees Celsius, and 72 degrees Celsius. As you may have noticed, these are quite high temperature. That's the reason why TAC is actually one of the best at performing this. All right. So once we have that, I think that's pretty much all that we can talk about PCR at this level. As you see again, this is just a very nice picture that we've actually borrowed from Khan Academy. Here it shows you again that during this PCR reaction, you have a DNA strand. Let's say it was like this, and here is your region of interest. From this region of interest, what you're going to notice is that we have, first of all, denaturation. We've broken the hydrogen bonds here. We've opened the strand at this temperature. Then we added our primers, and as soon as the primers were added, now extension was possible because probably polymerase is here adding those chains, and we've started with one strand and we've doubled the number, and we can repeat this about 25 to 35 times. And of course, this is a very important process in molecular biology because it may allow us to study a specific gene, to amplify, to study the expression, and the role and the importance of this is just countless. But I really hope that you learned at least something new from this small video, and thank you very much.